everybody. Welcome to the first day of this conference. This is great to see such a nice crowd here and more online. Excellent. So let me just go ahead and start. I'm going to talk about benefit risk considerations. And what I'm going to go do during this talk is go through some of the key issues and key considerations in regulatory decision making and their application during drug development. So I'll do this with the help of case studies. And I'll use two examples of clinical programs which were placed on clinical hold due to safety concerns that emerged during the development. So regulatory decision-making and benefit risk considerations, this can be straightforward, but many times this becomes a complex issue, primarily because this can involve several aspects, several issues, several considerations. Decision-making should really be based on clinical benefits, the risks associated with product use, as well as taking into consideration the disease context that the drug is intended to treat. Importantly, these decisions should not be arbitrary, and what's needed is really a consistent and systematic approach, and this is what's critical. So when it comes to benefit risk assessment, one thing to remember is that this terminology is typically referred to in the context of drug approval. And this is what I mean by this is that at the time of drug approval, when an NDA package comes in, there is usually substantial evidence for benefit or product effectiveness. And at the same time, there is evidence for product-related risk. And so making a benefit risk assessment becomes, is possible. It's possible at this point. That said, the similar general principles do apply to decision-making during drug development, that is, during the IND phase and even post-approval. So what I'm going to go through now is some of these elements, some of these basic elements which are necessary for the regulatory decision-making process. So first, let's look at benefit. As the name suggests, benefit denotes product effectiveness. And during the IND stage, what this means is there may be just some proof of activity data that may be available for this product. Risks, by this we mean product safety, and this includes human safety data, toxicology data, as well as other non-clinical data that may be taken into consideration when considering human use and human safety. Class-related toxicity concerns may emerge, and there may be consideration for risk minimizing strategies as well at this point. Now, it's important to take into account the activity as well as the safety profile in the context of the disease for which the drug is being developed. So in this point, we're talking about the nature and severity of the condition that the drug is intended to treat or prevent. And linked to the disease condition and the disease context, we have to think about the medical need, and we have to think about the attributes of alternative available therapies. So is this a condition for which there are approved drugs and approved therapies in the market? Is there an unmet medical need? And for example, a few years ago, there was the Ebola outbreak. This was a global crisis. And Ebola infection is something that we all know. It's a life-threatening disease. It's something which is associated with high fatality rate. There are no approved medications or therapies for Ebola. And this Ebola infection, this represents an unmet medical need for a life-threatening condition. And when you consider the benefit risk assessment, you take that into consideration versus another condition which is non-serious and for which there are multiple approved therapies. And with all these risk-benefit assessments, despite having activity data, despite having safety data, often there are areas of data gap, or data uncertainty. So there are areas where you wish you had more data. You would like the sponsor to give more data to better understand the situation, but there is some level of uncertainty. And at this point, the division, the reviewers, as well as the sponsors have to think about whether it's possible and whether it's reasonable to obtain additional information to respond to the data gap. And sometimes when there is uncertainty, you have to weigh the level of uncertainty into the overall equation when making formal benefit risk assessment and making the decisions. And there may be many more other considerations which are specific to the particular drug or the intended indication, but what I've outlined so far are just the overall broad and general issues and elements that have to be pulled together while making your benefit risk assessment. So moving on to case studies. The case studies that I will present are for antiviral drugs, and I work in the division of antiviral products. We review products which are intended for viral infections and conditions, not just Ebola and HIV. We review drugs for hepatitis C as well as influenza. And one of the issues I do want to mention is that some of the aspects and nuances which I'll discuss in the forthcoming cases may be very specific to antiviral products and may not be applicable globally to programs reviewed by other divisions in CDER. So just keep that in mind. So the first case study, and this is an example of a clinical program that was ongoing, but a safety concern was identified during phase 2, while the phase 2 clinical trial was ongoing. And what happened was the safety concern was explored further. The review division and the sponsor, we looked at all the information and we made an assessment. And the decision that was made for this particular IND was to place it on partial clinical hold. The forthcoming talks will go into more details about what constitutes a partial hold versus a full hold and some of the more technical details. The point here is the thinking process, and I do want to point out here that this decision to place the IND on partial hold took into consideration that there are two distinctly different HIV subpopulations which the sponsor was trying to develop the product for. And for those who are not familiar with HIV infection, the treatment-naive HIV-infected patients are treated somewhat differently from treatment-experienced infected patients because treatment-naive patients have many more safer alternative therapies available and approved for them. Compared to that, treatment-experienced patients are those who have slightly more advanced disease and they have fewer treatment options. So that's the key over here. And let's just go through the case so you can understand how we went through the decision making. So this is a program that was being for an antiviral drug for HIV. The sponsor had submitted the requisite toxicology data to start their first-in-human clinical trial. Single and multiple dose clinical trials had been completed. A proof of concept trial had been completed, so data were available regarding activity of the product in the target population, and a 6-month clinical trial was ongoing. At this point, the FDA was notified of a serious adverse event, which is described here, and I'll let you go through this. So this is a patient, a subject in the treatment trial. This is a treatment-naive subject. You can see increases in ALT, AST, bilirubin. These are hepatic liver parameters. A liver biopsy is performed, and there is a diagnosis. And here is the same participant, the same subject, this liver parameters, hepatic lab parameters, and you can see baseline values, which are normal. Treatment is started, steep increase in values, and then once the treatment is stopped, decline in values. So just go ahead and ask you a question, and for those in the room, please just show of hands. Do you think this case represents a safety concern or significant hepatotoxicity? Excellent, perfect. So, so this is a case of drug-induced liver injury, and let's just walk through what the next steps are and how ultimately the decision was made to place this IND on partial, not full, partial. So this is drug-induced liver injury, daily hepatotoxicity. This is a huge concern, and all development programs are, this is something that all development programs should be obviously monitoring very carefully in their trials. Now, DILI may or may not be dose-related. It may not be observed in animals, and it's somewhat unpredictable for most drugs. Generally, the incidence is relatively low, and it's often discovered post-approval. Coming back to our case, for this clinical program, clearly the agency was very concerned when we saw this, and what we did moving forward was we spoke to the applicant and discussed with them in detail that particular case, the Sentinel case, and we asked and requested that they look through the entire clinical program for additional cases which had a similar profile. So we made a request for like cases which had been observed in the clinical program. 10 such cases were identified, of which a total of 4 cases were considered relevant. And this included one case of de-challenge and re-challenge. And we also noted that the cases occurred at all doses of the drug. So there wasn't a particular dose that was associated with toxicity. It basically was throughout the sequences. So this slide goes through the cases, the sentinel case, the second case, which was at a lower dose, a third case, which was more compelling because it had de-challenge and re-challenge information, and a final fourth case, which was somewhat confounded because of prior history and some confounding issues like alcohol abuse. And I'm not going to go into detail, but I show you this primarily to emphasize that the clinical reviewers then really start looking at all the information and all the cases, and we meticulously go through everything and every little aspect and every little enzyme level that goes up. And that's when we all sit down and we go through along with the sponsor and discuss and understand what is going on with this particular drug. Is this drug-induced toxicity, or was there something else that could explain the finding? This is extremely concerning, and here what you see are some of our some of the tenets or elements that we look at as we try to assess the causality in this case. And we, like I said, we look at time to onset, look at information related to de-challenge and risk factors, and we also go back and look at the track record. So does this drug belong to a class of products which are known to cause hepatotoxicity or have some signal or some kind of a raise red flag for drug-induced liver injury, and etc. So in case of this example, we determined this was in fact drug-induced liver injury. It's a very serious safety concern. And at this point in the development program, like I mentioned, they were in phase 2 and they were ongoing trials. And we looked at the entire clinical program, and there were about 300 patients who were treatment naive, who had been exposed for roughly 10 to 12 weeks, and about 50 treatment experienced patients had been exposed for a median time of about 30 days. And as we started thinking about this some more, clearly this was a safety concern. However, when you broke it down by subpopulations, we looked at the incidence of these grade 3, grade 4 liver abnormalities, and we noted there were none in treatment experienced patients, although much fewer of them had been exposed. And as we started thinking about this, and this, mind you, this is a time when there were very few HIV drugs or antiretroviral drugs that had been approved. And even the ones which had been approved, majority of them really were for treatment naive patients, and very few drugs were available for treatment experienced patients. And so keeping that in mind, we made a determination that the IND would be placed on partial hold such that treatment experienced patients could continue to receive clinical dosing in clinical trials. And I'm not going to, again, go through this busy slide, but I show it here to emphasize all the different aspects and nuances that go into the decision making. We looked at the intended indication, what's HIV infection, the available treatments, and we broke it down by subpopulation. And we noted that the treatment experienced population was the one that had a relative unmet need. There was proof of activity data that was available for this drug, which was great. There was a concern for hepatotoxicity. Nevertheless, this was a concern that could be managed by very close, careful monitoring, by including some stringent criteria with clinical dosing in clinical trials. And the ultimate decision was to place the IND on partial clinical hold such that dosing was allowed to proceed for treatment experienced patients. So before I go on to the second case, I'm just going to pause here to see if there are any burning questions. So the second case study, and this again is an antiviral drug. This is a somewhat interesting case. This is an example where toxicity or the safety concern was identified during phase 3, but this was toxicity which was identified in a chronic toxicology study while the clinical program was ongoing, and we decided to put the IND on full clinical hold. So full clinical hold basically is where all clinical studies under the IND are prohibited until the hold issues are resolved. What's interesting about this example is that after the sponsor and the IND was placed on full clinical hold, the sponsor came back to us and they had revised the development program. So they revised the development program to focus on a specific subpopulation which had a medical need. And after review of the data and after consideration and deliberations, the full clinical hold was converted to partial clinical hold. So let's go through this one. This IND was for genital herpes. Genital herpes, it's a common sexually transmitted infection and manifests as localized painful sores, skin lesions, and it's not really viewed as a life-threatening condition. It's bothersome, of course, but not life-threatening. Now, herpes can vary. The clinical disease can vary depending on the host immune status. So in immunosuppressed or immunocompromised patients, the skin lesions are much more serious, much more severe, and they're extensive ulcerative lesions which last a long duration. And this is typically in those patients who are on immunosuppressive treatments or have HIV infection. So a small group of patients can have severe disease. Herpes treatments, a variety of treatments that are available on the market. Most of them are reasonably well tolerated with some concern for renal safety. That said, for immunocompromised patients, the ones who have that sicker, more severe disease with resistant or severe herpes simplex infection, they have fewer available treatments, and the ones that are available have significant toxicity, including boxed warning. So the investigational agent, so this clinical program, the investigational agent was being developed for treatment of genital herpes in immunocompetent adults. The drug itself, an antiviral drug, had a novel mechanism of action and it exerted antiviral activity at a site that was different. So very unique mechanism of action, which was an advantage. The toxicology package which had been submitted for the initial IND was not concerning per se. The new IND was allowed to proceed, and it was during phase 2 that we were notified of animal toxicities in the chronic toxicology study, which had not been identified previously. Now, these were severe drug-related toxicities, and they resulted in early or unscheduled animal sacrifices. So this was concerning. And these are some of the considerations that the reviewers, including the toxicology reviewers, think of and take into account when they're gauging how serious the toxicity is. And I'm not going to belabor on this because tomorrow, the first session, I believe, is by Dr. Gantus, and she will cover some of these toxicology issues which are in her talk. So I'm not going to go over this in detail. And here's the conclusion. Severe toxicities. The drug is being developed for genital herpes. There are approved treatments out there. We basically put the IND on full clinical hold. So again, no studies, no clinical dosing can proceed. And although there was some clinical safety that had been obtained in the ongoing clinical trials, the extent of the toxicity profile, the extent and severity of these animal toxicities was sufficient to say, let's just stop development. So this IND went on full clinical hold. And after the sponsors put on full clinical hold, what we do is usually communicate the reason for hold in a full clinical hold letter. The letter includes not only the reason for hold, but also provides information on how one can resolve the hold. So, often with very serious toxicities, it's very difficult, very challenging, and there may be very limited options for the sponsors to resolve the hold issue. And especially if there are non-clinical toxicity findings, the sponsors can try to demonstrate that the toxicity is specific only to animals and not necessarily to humans. It's not relevant to humans. They can try to demonstrate that it was a study-specific issue. That's why there was a toxicity that was observed, and it was not necessarily relevant to, again, human dosing, or they can try to identify a population for which the benefit-risk assessment would be favorable. And in this case, just looking at the serious nature of the toxicities, we also advise the sponsor to consider revising their development program and perhaps trying to identify a population for which the risk-benefit profile would be favorable. And we thought this was probably the only feasible approach. And pretty much that is what happened. The sponsor did come back to us, and they had some additional animal toxicity information trying to explain the mechanism by which this toxicity developed in animals. But again, their ultimate revision was to change the population, and they decided to target patients who had immunocompromised disease and had resistant virus. So again, going back to the disease and background, keep in mind that immunocompromised patients, they develop much more severe disease. They have few treatment options. And this was an investigational agent which had a unique mechanism of action, so a unique way of targeting the viral enzymes, and it had a potential pathway to circumvent the resistance pathway and toxicities. That said, even with the revised patient population, the review division was quite concerned, extremely concerned, and we decided that it may be possible to dose these patients. However, it was important to carefully consider what the acceptable dosing would be. So we decided to limit the dose as well as the duration of dosing based on support from clinical trials conducted to date. So this is a situation where one was able to leverage some of the information from the clinical data that had been gathered and somehow try to create a path so that the product could move forward, primarily because there was a need in this particular niche of immunocompromised patients who had resistant virus. There was a need in that population, and there was some clinical data to support moving forward. So the sponsor did consider dosing for the revised population. They did agree to limit exposure to the defined dose and duration. We all agreed that although there were some safety findings and there was some safety data available from the clinical trials, those clinical trials were actually conducted in immunocompetent patients. And this is a slightly different group of immunocompromised patients. So there was still an overarching concern for product safety. And the clinical dosing had to proceed very carefully. Again, very stringent toxicity monitoring criteria, very conservative toxicity management plan, interim stopping rules, et cetera, and stopping criteria within the trial. So again, the trial could proceed in these sicker patients, in these immunocompromised patients, but with very careful monitoring to ensure human subject safety. And this is your benefit risk assessment. Again, we'll not go through each of these points in detail, but I do want to impress upon you that there are several tenets that we take into consideration, and it's clinical, it's regulatory, and it's very integrated when you think about toxicities and toxicology and clinical pharmacology, safety data. It's kind of all gets pulled together into the ultimate regulatory decision. And with that, I'll just end my talk and just summarize. I think the bottom line is that the context matters. We talk about benefit, we talk about risk, and we have to remember when a drug is being developed, it's being developed for use in a particular population for a certain disease condition. So context matters, safety issues and safety concerns are not assessed in isolation. What we really are looking at is the totality of data. I'm not going to go over these points again. I think you get the flavor from the two examples that I've given you already. And really these are the benefit risk considerations. This is something that's done during the approval process, but also during the IND stage as well as post-approval. So the same thinking process and the same approaches that are, that sort of are used during the entire product cycle. So with that, I'll end. I'll take any questions, and thank you so much for your time.
Okay, if you have any questions for Charu, you can either come up to the microphone or I can run the other microphone to you. So a show of hands. Not running anywhere yet. All right, think about it. In the meantime, let's take an online question, please.
Does the FDA use a specific program or tool for signal detection and risk assessments?
So do we use any kind of tools for detecting safety signals or safety concerns? Tools, so I guess it would depend upon the type of safety concern there is for drug-induced liver injury, for cardiac, I mean, there are a variety of tools that people are thinking of and developing. I'm not aware of one single tool that's sort of like pan-sensitive for all kinds of safety issues, but I think there has been a lot of work that's going on trying to look at certain ways that one can predict or project or screen patients, or even think about which patients are more at risk for drug-induced liver injury or for cardiac injury, et cetera. And a lot of work is being done regarding that, but I'm not aware of a tool that's been validated. Let's put it that way.
Okay, and we have our first question here in the room. Please, sir.
In the case of developing a drug for primarily a pediatric population, would you recommend, if it's going first in human in pediatrics, would you recommend starting with a phase 1 adult study, or would you recommend going directly to the pediatric population, even if it's a rare disease?
Even for?
A rare disease.
Right, so pediatric dosing, I guess the first question would be whether the disease condition is something that's only observed in a pediatric age population, or it's something that's across all age groups, and you could have adult data. So if a disease is only something that's seen in pediatric age patients, then there's no room for adult trials, right? However, I can give you the examples of our division where we've seen, for example, for HIV, it's a disease which it manifests across pediatric and adult age groups. And in this case, the adult data are very useful. If there is adult efficacy data and adult safety data, that's extremely useful in using that and extrapolating to pediatric age group. So in that scenario, you have adult data, and that can be leveraged and can be used to support pediatric dosing. You'd still need pediatric clinical trials. First in human trial would be adult. With pediatric dosing, and again, pediatrics is an entirely separate field with its own rules, regulations, and laws, but I think it's important to understand the prospect of benefit. And a lot of time when you have adult data and you have adult efficacy data, then the prospect of benefit or activity has already been established for that particular drug. So that's very useful moving on to the pediatric population. However, if the condition itself manifests only in the pediatric age group, then clearly you have to begin with pediatric dosing in that age group.
And let's go to our next online question.
Is clinical hold information, in parentheses, which IND and why, for example, copies of the clinical hold letter, available under FOIA or any kind of compliance database?
I actually don't know the answer to that. I think Maureen, would you happen to know the answer?
Sure. Is clinical hold information, which IND and why, for example, copies of the clinical hold letter, available under FOIA or any kind of compliance database? And the answer is no, because it's protected information. If the company chooses to share it, they can, but as the FDA, we cannot.
Oh, as the FDA, certainly, yeah, that's proprietary.
Okay, any other questions in the room? Next question online, please.
Okay. Is safety studies necessary when the API has a long history in a known application, but the study is used to determine new effects for the same application?
Great, so sounds like the API there's experience with the API in another group of patients, not necessarily the patients that are being evaluated for the intended indication. So I think safety, if you have human safety data, that's very useful. Whether that's sufficient for product approval for a new indication, that just depends on a case-by-case basis, but it's useful to have supportive safety, which even if it's from a slightly different population. I mean, like I said, the risk-benefit assessment varies, and something that may be a safety concern, that may be a showstopper for a certain population, may not be a showstopper for another population. So it all depends. It's hard to answer that question without knowing the details, but certainly having safety, human safety data is helpful.
Okay. Do we have another online question, or any in the room? Online?
We have one last question. When moving to a DSUR from an IND annual report, do you include a table of AEs or only SAEs?
I believe there's a talk
Could you
tomorrow
repeat
that
that
goes,
question for me a little bit?
Oh, do you want me to repeat?
Yeah.
The question is about DSURs and annual reports, and what happens is that initially sponsors submit annual reports annually, and then later on they could submit DSURs, which may be less frequent. I believe it's SAEs which are included in DSURs, but I may stand corrected on that, and I think there is a talk tomorrow on this which would go into details. I don't know if there is somebody here who can, or if somebody can help answer that. Yeah, I believe it's SAEs only, serious adverse events only, but I could stand corrected.
And that sounds like whoever asked that question online definitely needs to tune in tomorrow. Okay, can you please help me thank Charu for coming and talking with us today?
Thank you.